Good afternoon. Good morning, everybody. Not afternoon, but good morning. Okay. Um, let's see. We finished the nervous system on Tuesday, and we're going to begin the second communication system in the body. That's the endocrine system, right? And in terms of today, I'll lecture until 9 o'clock-ish. Then I'll pass out your quiz. Okay, so this is quiz 3. It covers the nervous system. Consider it preparation for next Thursday's exam. Okay, so remember, next Thursday's exam will cover, I figure I'll take a moment or two and tell you what it's going to be. So the way the exams work, you have one question per system. The introduction is considered a system. So this exam covers the introduction, the nervous system, and the endocrine system. So therefore, there will be 3 questions. There will be 3 pages to the exam. Okay. Now, the way that works is in years past, I would ask a single question and you would take and fill up a page with your answer. All right. However, over the years, I've gotten a little bit soft. So instead of doing that approach, that was when I was where you were at, and I took physiology, I took advanced physiology where we'd have a class on the heart, class on the lungs, et cetera, et cetera. The standard exam for me, you know, those little exam books, those 20-page exam books, the instructor would write the question on the board, they'd give you the book and you fill it up. Okay. So in other words, you'd have to incorporate everything you know to answer the question. I've modified that considerably. We don't do that. So what you'll do is you will have a page that will have a list of questions that correspond to the introduction, a page that will have questions that correspond to the nervous system and the endocrine system. All right. They're going to be short answer / essay questions. All right. So fairly easy to understand. In other words, I don't ask, you will be asking answering specific questions. All right. I've given you examples of those questions on Canvas. Also, by answering the quizzes and filling out the quizzes, those help you as well. All right. So that's for next Thursday. Let's concentrate right now on moving to the endocrine system. All right. Now, when you guys get through, I'd probably ask you, do you have a favorite system? Some students do have a favorite system. Some people don't. And then others, contrastingly, have the systems which they like the least. All right. Now, my training is in endocrinology. That's how I was trained. So I tend to obviously have a soft spot for the endocrine system. Is what I do. It's what I've done for decades, what I continue to do until I retire. All right. Now I'm an engineer by application. All right. So what we want to do is I'll talk about this particular field. And what's kind of interesting is this, the field of endocrinology dates back to 1905. All right. The Endocrine Society was formed in 1905. It's still going strong. I've been a member for 50 years. All right. So it's been around a while, and there's been a lot of changes over that. And what's interesting is and it isn't a dead field because we're discovering new hormones every year. It's just like the field of physiology in general. It is an advancing, emerging, evolving field. Now, the stuff that I teach you for the most part is established science because this is an introductory course. But every once in a while, I'll throw in some new discovery, which kind of flips things on their head a little bit. Okay. Now, in the field of endocrinology, as we'll talk about momentarily, there is a historical aspect, and then there is the modern aspect. Okay. The textbooks that are in your physiology textbooks, they are 30 years out of date in terms of endocrinology. Okay. And terminology. I will correct that here. All right. So what I'm giving you is the most up-to-date information. All right. If what you read in the textbook differs, it's wrong. I'll tell you that right now. Okay. Most textbooks, by the time they're published, they're already 3 years out of date. That's just standard protocol. Okay. You can't stay up to the newest cutting edge stuff. All right. Or major changes. So we'll use that as an introduction, and I'm going to begin. So I've already told you repeatedly that you are a multicelled organism. Now, you can do your calculations however you wish. In some cases, I've asked homework questions where I ask you to calculate the number of cells in the body. It's all based on depending on what your assumptions are, because no one has ever disassociated a human person and then counted all the cells. That's just not physically possible. All right. And it's not particularly relevant here. You can pick whatever number. We'll say 75 trillion cells. That's the number I tend to use. All right. Now, what's critical is all those cells have to communicate because you act as a functional individual. That means that all of those 75 trillion cells in some way or another have to communicate. We've talked about the neural system and how that plays a role. Now let's focus on the endocrine system, right? And I've also, when we were into control systems, I said physiology is nothing but this huge conglomerate of interacting control systems. And one of the primary hallmarks of those control systems is the endocrine system, because that's basically all it is you're going to see it's nothing but control systems, right? They're controlled by hormones, these chemical entities, which I'm going to spend some time. So basically, the endocrine system in its simplicity is cell A talking to cell B via some chemical intermediary. That's basically the endocrine system. Right? So let's get some definitions. I'm giving you the most up-to-date definition. This comes from Becker. This is a text in Endocrinology, 2001. It's about this thick. Okay, I have a copy in my office. And basically, it defines, this is the modern definition of endocrinology, not the definition from the 1930s, not the definition from the 1970s. This is the definition as of now. Basically, the study and communication and control within a living organism by means of chemical messengers that are synthesized in whole or in part by that organism. Okay? But this isn't the complete answer. All of you have heard of chemical disruptors in today's society. You've heard about micro and nanoplastics. Some aspects of those are endocrine disruptors. So that means they can piggyback on your endocrine system and screw things up. We've known about these chemical disruptors for decades. All right. There's water bottles here that are plastic based. When you buy them, they say BHA free. Why? Because BHA is an endocrine disruptor. Right. So even though that's not part of the endocrine system, it can have impact on the endocrine system. And I will talk a little bit about that at the end of this series. All right. But basically, this is what endocrinology is. Next, those chemical entities are referred to as hormones. Right? And they are responsible not only for cell-to-cell communication, but for communication within a given cell. So I want to talk about the history of endocrinology and what it is and how it's evolved over time, and some aspects of which we haven't all changed. So the original definition of hormones dating back to 1905 and the origin of the actual field is that an agent that can excite or arouse. That was the original definition of a hormone. Right? Now, during the next intervening 30, 40 years, discovery of new hormones was brought about. Now, the basis of the discovery was blood. Why blood? Because it's minimally invasively, it can be taken without major threat to you, and it can be analyzed and so on. So the field of endocrinology was this. The field of endocrinology evolved by you have a chemical entity, you take away surgically some particular organ, you have a loss of this chemical entity, then you have some sort of pathology that results. You add that entity back and you recover function. That's how new hormones were discovered up until around the 1930s and 40s. Okay, so that led to the definition of hormones being the classic definition: a chemical that was secreted by a specific gland that then entered the bloodstream, traveled to some distant part of the body, and elicited a function. All right. So, for instance, a hormone was released from my pituitary gland, which is go up to the roof of my mouth and back a little bit and up about 3 centimeters. That's where that is. It's in your brain. And it releases a hormone that enters the bloodstream, travels to my big toe. All right. That's a classic definition of hormone. Right. Now, that held through until the 1970s, especially the 1980s. And that gradually was incorporated into the modern definition is that a chemical entity is secreted or produced by specific cells or cell types and can then have action. Because what happened in the 70s and 80s, we found that, wait a minute, hormones are all over the place and they don't have to be secreted by a specific organ. Every living cell in your body secretes a hormone of some form or another. Multiple hormones for the most part. So all those 75 trillion cells in your body, the living ones, they can be secreting hormones, right? So this is the modern definition, and I'll do some more clarification as we go along. Now, the classic endocrine system is based on, when you see the anatomy here, they will show you specific glands. So you go up here into the brain, we have the hypothalamus, we have the pituitary, which consists of the anterior and the posterior pituitary. We also have the pineal gland. Those are all within your brain. We drop on down a little bit lower down here. We have the thyroid gland. We have the parathyroid glands here. Then we drop on down. We have these two pyramid structures that sit on top of each of your kidneys. They are your adrenal glands. They consist of a medulla and an inner cortex, okay? An outer cortex, all right? We then can go down in terms of reproductive physiology. We either have testes or we have ovaries, and they produce particular hormones, all right? Then, obviously, with the new modern definitions, a lot of hormones that have been discovered in the last 20 to 30 years are having to do with the digestive system, all right? Gastrin and all these kinds of things. GLP-1, you know, those are big drugs. Those were not discovered. Those were discovered just a few decades ago. All right. So basically, they have big impact, and they're not produced. Well, they can be produced in localized tissues, but for the most part, yes, they can be produced predominantly in one particular tissue or not, but they're not limited to that. Every living cell can be a source of a hormone. Now, what we talk about when we talk about the field of endocrinology is cell talking to cell. How does a cell talk to a cell? That's what I want to focus on right here. And I want you to understand that, for instance, let's start down here. This is where we have a specific gland or a number of tissues, cells within a tissue. They secrete a hormone, it enters the bloodstream and travels to some area. That was the classic decades ago. That's 80, 70 years ago. That's what endocrinology was called. It's no longer that. It's all of this. So this is no longer called an endocrine. This is not an endocrine form of communication. It's called hemocrine, which means via the blood, or this is the most modern term, which dates back less than 20 years, which is telecrine, which is simply means long distance. All right. So if it enters the bloodstream, it is referred to as hemocrine or telecrine. All right. Now, all this other stuff, let me talk about because it's critically important. Because it's all dealing with cell-to-cell communication. It depends on which cell is talking to which cell. In this case, some cell that's up here is talking to a cell in my big toe. Long-distance communication, all right? Hence telecrine. Now, let's start up here. Endocrine, which means that the hormone actually doesn't have to leave the source cell. The source is synthesizing it. It doesn't leave. It stays within the cell and signals the cell from inside the cell. That's called endocrine communication. Not cell to cell, but cell within cell. Next, if we drop down to right here, we have autocrine. This form of communication dates back to 1978, right? And basically what this states is that a cell releases a hormone, it enters the interstitial space, and then signals back on that source cell. All right? That's called autocrine. So the cell produces the hormone that then tells that cell what to do. Hence the word autocrine. This was first proposed back in 1978 to explain the source of cancer. The hormone that fulfilled this role was something called transforming growth factor beta. The name is that. It means by transforming, that means it induced normal cells to be cancer-like. Yes, TGF beta. And at the time, they proposed that they had solved cancer. Where it came from, bullshit. All living cells secrete TGF beta. It plays critical roles throughout development as well as disease states, but it doesn't matter. This is a form of autocrine communication. All right. This already blew the water out of the old classic definition as early as 1978. Right now, during the 70s and 80s, there were groups of hormones that were really being actively discovered called growth factors, which is another area I've been working in since the mid 80s. All right. So things like TGF beta is a growth factor. It's like growth factors or growth factors, etc. There's a huge array of them, and they don't act as traditional growth factors, right, for the most part. Now, we also have something called paracrine communication. So paracrine communication means that communication is limited to cells within a tissue. That means local control. All right. So that means I'm in a tissue. Here's a cell making the hormone. Here's the cell that has a receptor population and responds to that hormone. It's all within the tissue. All right. Now, if we look up top here, we have intercrine communication. We've already covered intercrine communication. We talk about gap junctions. The deal is that that is limited to very small molecules passing through very small holes. Now, I don't remember if I introduced TNTs or tunneling nanotubes, but those are bigger channels between connected cells, and they allow for much bigger things. Even things like mitochondria can be transferred, right? So that's still a form of intercrine communication because it's direct cell-to-cell exchange without dilution in the interstitial environment, right? Now, the next one down is juxtacrine. This form of communication dates back to 1991, right? We're still older than you are, but nevertheless, in 1991, in this case, what they found was, here are two cells. My thumb is the hormone that is attached to cell A. Here's my fist. This is cell B. While this hormone is on the surface of cell A, it can directly bind to the receptor on cell B, which means they have to be closely approximated for the hormone on the surface of cell A to work with a receptor on cell B. This is called juxtacrine communication, which just simply means short distance. Okay? That has to be a direct contact. The next one is a little bit more confusing. It's referred to as solencrine. What this means is I have a tube structure in the body. It can be a seminiferous tubule. It could be a blood vessel. It doesn't matter. We have a biological tube structure. There are cells that line that structure. They are releasing hormones that enter the luminal space. So those types of hormone, then from there, they can go wherever. That's called solencrine, right? Now, up here at the top right, we have sympathetic and non-sympathetic. Wait a minute, we discussed that in the nervous system, right? So basically what we're talking about, sympathetic, non-sympathetic. So, when a neuron A talks to neuron B, what has to happen? It has to release a neurotransmitter. A neurotransmitter technically is a hormone because it's a chemical entity allowing the communication between two adjacent cells, right? So we're already starting to see how the nervous system and the endocrine system are really intermeshed, as are most systems. And that's fine. There's nothing wrong with that. So basically, I will give you other examples of this interconnectivity as well. Because, hey, I just showed you the anatomy in a couple slides back where the brain was the source of hormones. That brain is the central nervous system. It's also part of the endocrine system. Okay. All right, so why do we have these? Why do we need these chemical agents other than what does it mean allowing for cell-to-cell communication? What are the functions of hormones throughout your body? Well, number one, reproduction. All right. We'll cover that. We're not going to cover that in the endocrine portion. We're actually cover that when we get to reproductive physiology, because I tell you right now, all reproductive physiology is nothing but endocrinology, because it is those hormones which make us physiologically female and male. Right? And we will spend a considerable amount of time talking about those reproductive hormones. But that's another story. Next. Hormones are critical for growth and development. You begin life as a fertilized ovum, a single cell. How do you get to 75 trillion cells? Mitosis and differentiation. All of those processes are directed and driven by hormones. Dropping on down, maintenance of the internal environment. This is everything from pressure in your blood. This is gases. This is glucose, sodium, potassium concentrations in your blood. All of these things are controlled by hormones. And as well as energy production, utilization, and storage. So basically all physiological functions are controlled by hormones. Now, the thing to remember is this. Where do these controls take place? All of the controls start at the cell level. So what we're talking about is a cell level effect. So what does that mean? Even though, for instance, let's say I'm going to stimulate your heart rate. We're talking about this heart is comprised of millions and millions of cells and very complex anatomy. However, controlling that has to begin at the cell level. That means a chemical entity, i.e. a hormone, binds to a receptor on an individual cell, group of cells, and it can change the functionality of that tissue, or in this case, that organ, the heart. So all communication, all endocrine communication is initiated at the cell level, right? And we will emphasize and discuss how that happens in this section. Now, at the cell level, what's being changed? All aspects of cell behavior. That could be you have hormones that tell cells to self-destruct, apoptose, or to survive. There are hormones that will tell those cells to undergo mitosis, make lots of daughter cells, and that's cell proliferation. There can be hormones that will tell a stem cell population to direct them down to become other some cell fate, maybe become muscle cells, neuron cells, blood cells, whatever. All right? Those are all controlled by hormones, as is all general type cell behavior, everything from cell secretory products, let's say a mammary epithelial cell and a lactating female, all that milk is produced by a mammary epithelial cell. Hormones control the functionality of that cell, and then that hence its behavior. Or we can talk about directing the migration of cells from this group, this area to this area. Those are cell behaviors. All of those are cell behaviors. Now, whether these functions are resulting it depends upon this. Number one, remember, everything begins at the cell level. So therefore, you have to have a responding cell type. What does this mean? If you have a chemical entity and it's communicating with a cell, first, there has to be a receptor for that chemical molecule. If that cell does not contain a receptor, the cell cannot sense or it cannot communicate. Okay? So that not only does there have to be a receptor, all of the post-receptor stuff, all the downstream stuff has to be there too. Okay? Because this hormone ligand, if you will, binding to the receptor just initiates communication. And it is a whole series of events that occur in the cell that will then result in these cell behaviors, which I mentioned in the prior slide. But all of these depend upon this responding cell type. It has to have the receptor, and it has to have all the machinery to respond to that binding interaction. Next, So that means you have a signaling molecule, the hormone, it has to bind to receptors. If I play with the concentration of the hormone, I increase the likelihood of it encountering a receptor, right? So as I increase the concentration of that hormone, I can expect it to have to change. It should have more of an effect because it's interacting, more likely to interact with the receptor population, right? Lastly, presence of other stimuli. There are thousands of hormones interacting with the cells in your body at any given time. There are other constraints as well. And the whole point is this, the take home is this, there is multiple interacting stimuli that can modify the communication between a hormone, a receptor, and a responding cell type, all right? But all three of these will determine whether a cell responds to a hormone. Now, I'm going to be stimulating a cell with these hormones. Now, the stimulus can result, what does that mean? So, the hormone has to bind to a receptor, but what happens after that? So, the receptor's job is to say, yes, there's a hormone. And then once this binding interaction occurs, something then is initiating post-binding. That changes cell behavior. These are some examples of what those things can be. Number one, maybe it alters plasma membrane permeability. We've already discussed this. This is the postsynaptic membrane of that on the synaptic cleft, right? It is a chemical gated channel. There's a receptor on this transport protein. When it's occupied by a neurotransmitter, which is also a hormone, what happens? It changes the permeability. It allows in the case of a chemical gated channel for sodium, it opens, that means sodium flows into that across a cell postsynaptic membrane down its concentration gradient. So it's altering the permeability of that membrane. That's just one example. There are many. Now, next, a hormone binds to a receptor. Eventually, the downstream effect is that's going to change the synthesis of new proteins within that cell. So that means that the DNA is going to be read. You're going to get messenger RNA translated to protein, new protein, novel protein production, right? Or maybe it can be whether else, okay? Maybe it blocks the synthesis of those proteins. It can also, the binding interaction can also result in the activation or inactivation of enzymes on the inside of the cell. This can then play some role in communication processes. It can induce secretory activity. I've given you the example of a mammary epithelial cell secreting milk under the direction and control of a hormone. Lastly, it can stimulate mitosis and other cell behaviors, all right? So basically, these are what happens after a hormone binds to a receptor within a responding cell type, who has the appropriate machinery to act on it. So what are these molecules, right? Basically, we divide them in two broad categories. They are amino acid based or they're lipid based, okay? That's it. So what you don't see, calcium is not a hormone. Sodium is not a hormone. These are ions. They are not hormones, though they don't have specific receptors per se. Okay. So what we do talk about are amines. These are basically amino acids. So we have melatonin, epinephrine, norepinephrine. Remember, we've talked about those. All right. They and we also have thyroxine that's produced by your thyroid gland here. T3, T4. So these are very small molecules. They and basically they are plastic hormones as well. In the case of epinephrine, norepinephrine, the catecholamines, they are produced by the adrenal medulla. These pyramid structures back here, they travel throughout the body, and they then are responsible for sympathetic stimulation of stuff. Okay, in terms of your heart, they will increase your heart rate. They will increase the heart contractility. They will also increase venous return. The net effect is to increase blood pressure. All right. Dropping on down, they can be peptides. Remember, you have amino acids. Groups of amino acids make peptides. Larger groups of amino acids make proteins. What's the difference between a peptide and a protein? If you take my class in the spring in engineering protein therapies, we define that. And it is an arbitrary definition. Actually, the FDA's definition is that it's anything less than, I think, 50 amino acids. Okay. But most peptides are probably less than 20 amino acids, and they can be as little as 8 or 7 amino acids. There is no clear definition, all right? And the FDA just made their definition sort of arbitrarily, but they needed to be able to say, this is a peptide, this is a protein. They just threw a dart at the wall and said, this is what it is. There's nothing new. There's nothing unique about that number that they use, all right? So, but you have peptides, and those can be oxytocin, parathyroid hormone, antidiuretic hormone. Don't worry about what they are. We will, those, for instance, we'll talk about all three of those later in the semester. All right. Also, proteins, these are larger. These can be continuous chains. They can be discontinuous chains of amino acids. So, growth hormone, insulin, glucagon, insulin-like growth factor, all of these are examples of proteins. Now, remember, there are thousands of these. These are just a very small representative samples. Now, next we have the lipid-based hormones, and they're based on fats, okay? So, all of them are the steroid hormones are all based on cholesterol. You've been told at some time that cholesterol is bad, bullshit. It isn't, okay? And if you don't consume in your diet cholesterol, your body makes cholesterol because it's necessary for life functions. In fact, over 80% of the cholesterol in your body is made by your body. So it doesn't matter whether you eat an egg a day or 2 eggs a day or anything like that. Now, if your body's producing an excess amount of cholesterol, that's when you have to worry about and bringing in additional nutritional and adding to that. But that's a different. We'll have that subject discussion later. So cholesterol is also the initiating chemical that all steroid hormones are derived from. Whether we're talking about testosterone, progesterone, and estrogen, these are the sex hormones, or cortisol, which is involved in metabolism. All right, and stress response. So those are steroid-based. Now we have another lipid-derived, which is the prostaglandins, right? But the key is that the key definition is from proteins up. These do not readily cross. They do not cross the lipid bilayer of the plasma membrane. These, because they are fat-based, do. Okay. So what does that mean? Where are the receptors for these hormones? It depends on the source of that hormone. If it is an amino acid based, that means it does not cross the lipid bilayer by itself. So, wait a minute. I need to communicate into the cell. How do I get that communication into the cell? That means the receptor has to be on the surface of the cell. I repeat, on the surface. So, those types of amino acid-based hormones will bind to receptors on the surface of the cell. Now, at that point, that's the binding modality, but then that initiates communication cascades within the cell. Hence, we can then communicate into the cell. But it has to begin on the surface of the cell. If, on the other hand, I have fat-based hormones, whether they are steroids, like estrogen and testosterone, whether they are prostaglandins, they readily diffuse across the membrane, lipid bilayer. So the receptors for those hormones are inside the cell. I repeat, they're inside the cell. And in some cases, they can be in the cytosol. In some cases, they can be actually in the nucleus. Okay, because if you go through the plasma membrane, you can generally easily get into the nuclear membrane as well. All right. Now, so what this is a critical aspect of intercommunication, depending on the nature of that hormone. If it's amino acid based, the receptors have to be on the surface. If it is fat based, the receptors are on the inside of the cell. Right? This is just showing the same thing. So is it hydrophilic or is it hydrophobic? If it's hydrophilic, the receptors are on the surface. If it's hydrophobic, it's on the inside. Okay. Now, this is simple tissue compartment. It's a group of cells. I want to use this to illustrate cell-to-cell communication, right? So everything that is outside of these cells is in the interstitial compartment, right? You remember those definitions I gave you in the introduction. Now, we have a mixture of cells, and I want you to note H and HR. H stands for hormone, HR stands for hormone receptor, all right? Now, remember, you have to have a responding cell type to respond to a hormone, right? So in this particular system here, we have HR. So that means that this cell can respond to a hormone H. However, this cell does not contain hormones, receptors, so it cannot, but it is the source of those hormones. So it's producing the hormones, it releases in the interstitial space. They can act on this cell because it has hormones. This communication from here to here is paracrine communication, which I gave you an example of, generally speaking, about 4 slides ago. All right? Now, these cells can't respond, but they can actually create and secrete. They can be the source of the hormone. These cells cannot create it, but they can respond. Now, we also have some cells that both have the hormone and the receptor. So they are capable of autocrine communication. Now, do not think that you are limited to one form of communication for any given hormone. The reason I'm showing you this is that, that hormone can come from actually 3 places. Number 1, that hormone came from here. It came from cells within that compartment, right? And they acted on other cells which did not produce that hormone. Or they produce it and they can act on it. So we can have paracrine, we can have autocrine function. Now, this is the interstitial compartment that communicates directly to the blood, the plasma. This hormone doesn't have to be produced in this tissue. It could have gotten here via telecrine communication. That means it got here via the blood. Then it's transferred into the interstitial compartment. Once it's in the interstitial compartment, don't care. The other reason I'm showing you this is that the hormones that are communicating within this tissue can have a local source, whether it's paracrine or autocrine, or they can have a long-distance telecrine source from the blood. All right? As the hormones are all the same. It doesn't matter where they came from. It just means that that's going to create concentration here. Now, as I increase the concentration of the hormones in this interstitial compartment, what am I doing? I'm increasing the likelihood of interaction between those hormones and their corresponding receptors. Hence, and so as I increase the binding to the receptors, I'm going to increase the resulting physiological changes, those cell behavior changes. All right? All right, so that gets us to these 2 basic concepts. So we've kind of talked in generalities. Let's zero in. So the factors that govern the magnitude and responsiveness to a hormone. All right. So basically, what are those factors that will govern the magnitude of response? All right. So concentration, obviously. I mean, I've already alluded to that. So as I increase the concentration of the hormone available to bind to those receptors, I'm going to increase the likelihood of getting a resulting function. Makes sense, right? Now, how do I influence concentration? Where does concentration in that past slide, the hormones that are in that interstitial space, where do they come from? They can be the result of secretion. So they can be, within whatever physiological compartment I'm talking about, they can be the result of increased cell production and release into that compartment. They can be if they are coming from a systemic source, that can be a result of basically, the rate at which it's delivered to that compartment via the blood, the telecommunication. And lastly, what is the rate of destruction? So source and sink. Everyone has heard of source and sink, especially those in chemical engineering. Okay, so you have a site that's producing that protein, but it's not just some inert component, right? Not only is it capable of binding to receptors, let's say, giving you an example of a protein hormone or an amino acid protein. Remember, I talked about how in the nervous system, in your synaptic cleft, you have these enzymes, and their job is to quickly destroy those neurocommunicators, right? So basically, the only way you maintain the concentration of those neurotransmitters within the synapse is the secretion from the presynaptic membrane. Every time an axon comes down, you get a release, a bolt release of these hormones, but they're rapidly destroyed. And then you have to have another action potential coming down and the more release and so forth. The thing translates to all biological compartments in the body. So concentration is basically a function of three things: the rate of production within that tissue, the rate of delivery from a systemic source to that tissue, and the loss or degradation of that hormone within that tissue, right? All three of these interaction things taken together will influence concentration. Now, next is the number of competent cells that have functional receptors. So you already know that. I've already explained to you that a cell cannot sense or respond to a hormone unless it has an appropriate receptor, as well as all the post-receptor machinery to act on that binding interaction. So that's what this is. That's what a competent cell is. A competent cell means you have receptors that recognize that hormone and can respond with a change in cell behavior, right? The third aspect is sensitivity of the target cells to the hormone stimulation. What does that mean? So that's several different things. So first off, we have a cell. If I increase the number of receptors on that cell, what does that mean? That means we are increasing the likelihood of it binding available hormones. So number one, the number of receptors per cell, or the number of receptors within that tissue. Number two, it can be the binding affinity. All right, so we have a hormone, we have a receptor. That means that there is this specificity between this action, the binding of this ligand to the receptor. This is a result of binding affinity. That's the relative affinity for these to form a complex and create a hormone receptor complex, right? So I can either increase or decrease the affinity of this interaction. If I decrease the interaction, that means that these are less likely to interact, or once they're bound, to stay bound. If I increase the binding affinity, this means that when these are randomly there isn't like there's a homing signal in this hormone that says go, 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 go to this receptor. The way this hormone encounters this receptor is by random chance. So we have this molecule floating around. Here's the receptor on the surface of this cell. And it's all a matter of chance Brownian motion, that this is going to encounter this receptor, right? So if I increase the number of receptors without changing the number of molecules of hormone, that means that the cells with more receptors on their surface are most likely to interact, right? Now, if I change the affinity, that means I haven't changed the number, but I've changed the binding affinity. By increasing the binding affinity, I will increase the likelihood of this chance encounter to have permanence. That means that they will stay bound, and as long as they're bound, they're stimulating the cell, right? Now, this ligand receptor interaction is only the initiating aspect of this cascade. There's a whole cascade of events inside the cell. I talked about this in the introduction slides, where you had a growth factor or a protein hormone binding on the surface, and on the inside you saw all these multiple cascades of communication systems that result in cell behavior changes. The cell has to have those. If it has more of those, it means that it can have a greater response to a lesser concentration of hormone. If it has less of those or doesn't have them, that means even though the cell can bind a receptor, it can't result in a change in cell behavior because the cell machinery inside isn't functioning properly. All right. There are many, many diseases that are caused by disruptions in these communication cascades. Right. And lastly, effector molecules. All of these means that, for instance, if let's say that it's to stimulate novel protein synthesis, but if I don't have the amino acids available for that synthesis, I can't create more proteins. All right, because they are a critical component to that process. All right, so all of these factors will influence the magnitude of the response of a hormone within a potential responding cell. Right. And that gets us to the next thing. So the next thing is the factors that cover the duration. So magnitude of response is not duration. Duration is just how long is this process, or how long does it take from binding to a final whatever that behavior may be. All right, so let's talk about that. So the duration can be the duration of the available hormone, right? That is available to bind to the receptor. So that means that what's the rate of, so that think in terms of here's a tissue compartment. So think about that as a source and a sink. What's the rate of secretion within this compartment corresponding to the rate of removal of that secreted product? So you can have a rate of release of this new hormone into the space, but what happens if the rate of destruction exceeds the rate of production? That means you're not going to have stimulation because you're not going to have available hormones to bind to receptors. If we have a situation where we limit destruction, that means we will continue to stimulate. Let's go back to the nervous system and that synapse and talk about that. I did not specifically talk about that in class, but there are toxins within that are meant to have some impact, and they can disrupt this communication cascades. All right, so here is a presynaptic membrane, postsynaptic membrane. This is releasing neurotransmitters. They bind to the receptor. There are these enzymes that rapidly destroy that. What if I give a toxin that blocks the functionality of these enzymes? What happens? Yes, they keep communicating. So that is an example of hormone availability. I have not impacted secretion, but what I have done, I've disrupted degradation. All right. Now, also, hormones that are communicating via telekrin function, they're circulating in the bloodstream. What's their rate of removal from the blood? This is completely independent of secretion. They can be removed from blood via going to the tissues, being filtered out by the kidneys, and they can be degraded in the blood. any number of things. How long do they stay in there? Because that's going to be a function of how they can then be delivered to the tissues, right? Both of these influence duration. Now, whether and not only how long is that hormone available to interact with receptors, but how long does the process of what is the signaling process doing? How long does that cellular behavior change take? Maybe it is essentially nanoseconds because it's a membrane permeability, talking about neuron, we talking about that chemical gated channel, that post synaptic membrane, or maybe it takes days because it's a proliferation effect. And the cell doubling half-life is, say, 24 hours or maybe 36 hours. So that means that it's going to be days before you see the effect. So the effect can occur in nanoseconds. It can occur in days. And if we translate that to tissue changes, it could take years, right? So all of these can be the responses. Okay, we'll talk this and then I'll stop. All right. So what I wanted to do with this one is illustrate that here is the receptor cell here. Here's the receptor on the cell. Let's say it's a protein. That means that some aspect of this receptor is exposed to the interstitial compartment because it has to be there in order to bind and interact with the hormone molecule, right? Then we need to communicate. We need to get that binding interaction to result in a change in cell behavior. That means we need to get this binding interaction translated into some form of communication into the cell, right? So there is two aspects to these types of receptors. There is the ligand binding aspects. That's the part that recognizes and binds with some given affinity to the hormone molecule. Then there is since these are transmembrane proteins, then there has to be a transduction domain, which then says when this binds, this is going to send a signal into the cell. So all receptors have two aspects. All these surface receptors have two aspects. They have an aspect which actually recognizes the ligand, and then one other part of that protein molecule, when there's this conformational change resulting from the ligand binding to the receptor component, it changes this transduction domain, which initiates communication into the cell. All right? Now, that binding domain has specificity. We've already kind of talked about specificity. That means it recognizes a hormone, but then not another hormone. So it has specificity. The transduction domain is specific. That means when it is activated, it will send a set of signals that results in a behavior X. Okay? Now, for a given, so if we look up at the top left-hand corner up here, we have two hormones binding to their own individual receptors. So when hormone A binds to its receptor, that binding moiety then is occupied, that then turns on that transduction domain, and we get the physiological response to hormone A. Same thing for hormone B. Now, I'm a molecular biologist, and I like to screw up stuff. So I molecularly switch the binding aspects of B and put it on the transducing aspects of A, and put that on the transducing aspects of B. All right? So what I've done now is if A then will not result in the physiological changes associated with the native receptor for A, it will result in the one for B, because that's where I'm initiating the transduction domain for B instead of A. The whole point of this discussion is so that you realize that the binding component is, even though they're in the same molecule, the same molecule. So we've got this protein molecule. What is different is that the ligand binding part is different from the transduction part. And I'm just using this molecular biology experiment to demonstrate that. All right. Now, physiologically, things happen as well. What does that mean? That means that I can have in the same tissue, I can have, it can respond to a given hormone, but that means the binding, it has to recognize that hormone, but the resulting change in behavior is different because of the responding cell type. Remember, you have the binding and then you have the downstream stuff that happens. So we will get into the details of this more so when we get into the kidney. But in the kidney, there is a structure called the tubule. And there are epithelial cells that line this tubule. Depending on where you're at along the length of this, all of the cells will respond to a hormone. Here we call a PTH, parathyroid hormone. However, depending on which cells, where the cells are, they're all epithelial cells, but in this particular case, when the hormone binds to the receptor, because the intracell machinery is such, it will result in the activation of vitamin D3. Down further in the tubule, it does not have this intercell machinery. It has different machinery where it will alter the transport of calcium. So the point here is this: even though we have the same hormone binding to the same receptor part, the downstream stuff, the intercellular machinery, is different. So therefore, the behavior changes will be different and reflect that. So I can use this, if I can have the same hormone resulting in different physiological effects, just depending on if that is active inside those cells. All right. Okay. With that, we're going to stop because I need to give you your quiz.